Mr. Bonney here with another biology lesson, this time on protein synthesis. Protein synthesis is also called the central dogma, kind of an odd term, but that central dogma really refers to the central idea or main idea that we see in biology or in living things. And it's really a two-step process, and that process is going from DNA into RNA, and then from that RNA into proteins. That two-step process is first transcription, which happens in the nucleus, taking DNA, turning it into mRNA or messenger RNA, and then from that mRNA, messenger RNA, into proteins in the process of translation, which happens out in the cytoplasm with help from a ribosome. So a couple things to remember before we dive into the process is that we're going to be looking at genes here. And a gene is a sequence of DNA that has all of the nucleotides necessary to make one protein. So a gene is several nucleotides long, and it has all of the code needed to make one protein. On average, human genes are around 230 base pairs long, nucleotides long, and each chromosome themselves has several genes on it. The bigger chromosomes have more because they're longer. Chromosome 1 is a couple thousand, like around 2,000 genes on it. And then the smaller chromosomes will have less genes on them. But there's several genes on each chromosome. And when we're going to make proteins, we only use that one section of the DNA that's required to make that protein. Remember that one gene is equivalent to one protein, which is also equivalent to one trait. That'll come up again more when we get into our genetics unit. So here's our first step in the process, this is being transcription. Again, the idea here, the overview, is that we're going to take DNA and we're going to turn it into RNA, which is a close chemical cousin of DNA, and we'll get into the differences of that later. And that M, that little M in the front there, stands for messenger RNA, because transcription, if you kind of look at the root of that word, means to transcribe. To transcribe means to write a message. And that's essentially what's going to happen here in this first step. We're going to make a message from the DNA. That way the DNA never has to leave the nucleus. It can stay nice and protected inside of its nuclear membrane, and we can send the mRNA out into the cytoplasm, and it can be used over and over and over again. This keeps the DNA safe, so the DNA doesn't get damaged, and then therefore the cell doesn't have to incur any sort of damage or injury, anything like that. And again, all this happens inside the nucleus because, again, that's where we find the DNA. So we're going to take a look at the overview of the process of transcription here. So I've got a piece of DNA, and it's going to get unzipped, and it's going to get unzipped by helicase again. And then a new enzyme, RNA polymerase, is going to come in, and it's going to add in RNA nucleotides that are complementary. So again, we see that the G is complementary to the C complementary to G, A to T, but now we've got the new addition of U, which is uracil. Uracil is going to replace all of the thymine in this new piece of RNA. It's also important to note that we only use one side of the template strands here and not both sides like what we saw in DNA replication. This now completed piece of mRNA is going to break off and exit the nucleus through the nuclear pores. The DNA, the section that we use, the gene, is going to come back together so it can be reused. Again, so now we look at the differences between RNA and DNA. RNA is going to use uracil instead of thymine. The other three bases are all the same. Adenine, guanine, and cytosine still remain the same in both DNA and RNA, but uracil has now swapped out thymine, so you'll find no T in RNA. Another difference is that RNA is single-stranded as opposed to the double-stranded double helix structure that we see inside of DNA. And then finally, the sugar that's used in the sugar phosphate backbone of RNA is different. They use the sugar ribose, whereas DNA used the sugar deoxyribose. A reminder for some things on proteins before we jump into the last step, which is translation and the production of proteins, is what they are and what they're made of. Proteins are made of many amino acids held together by peptide bonds. So in this image here, you can see some individual amino acids, and then they're all held together in a chain with peptide bonds, but those aren't a protein yet. They need to fold up and become a three-dimensional shape before they're actually considered a protein. Remember that those peptide bonds are made in the process that's called dehydration synthesis, and then also there's 20 different types of amino acids. Finally, remember that proteins are really here to do cellular work. So when we talk about that the DNA controls the functions of a cell, it does so through the production of proteins. There's proteins that do digestion of food. There's proteins that work as antibodies that fight off foreign invaders. Proteins can be used to build muscle. Proteins are useful in copying the DNA and then making more proteins. Proteins do a lot. They do any and all cellular work. And so this process of making proteins ultimately is essential for life so that all of these things can be accomplished. All right, so again, let's jump into translation here. Translation, the overview of it, we're going to take that mRNA that was just made in transcription, and now we're going to turn it into proteins. This process happens in the cytoplasm with help from a ribosome. So let's take a look at this process now. So in this image here, first we see at the bottom of the screen, we see that strand of mRNA that we just made in transcription. Also around it, you see a ribosome, that's that pink blob. And what the ribosome is doing is it's reading the mRNA strand 3 letters at a time. And 3 letters or 3 bases on an mRNA strand is what we call a codon. So this first codon that we see the ribosome reading is AUG. And in fact, the first codon of every protein and every living thing is always AUG. Some of the other things that we see out here inside of the cytoplasm is we see this tRNA. Now the tRNA is what it stands for transfer RNA, and its job is to transfer the amino acids to the ribosome. So some things you'll notice on the tRNA is that it has a code as well. This first one we're looking at here has a code of UAC. That UAC is an anticodon that will match up with the first codon. A bonds to U, U bonds to A, G to C, so on and so forth. And then again, attached to the outside of all of these tRNA are all different types of amino acids. Remember, there's 20 different types. We just have a few that are showing here. So again, what's going to happen now is this first tRNA is going to bring in its amino acid to the ribosome. Once that's done and it's in place, the ribosome will then shift and it will read the next codon. The next codon in this case is CCU. So what's going to happen now is the next tRNA is going to bring in the next amino acid. From there, a peptide bond gets formed between those 2 amino acids. So now those 2 amino acids have been linked to each other. The ribosome is going to shift again. And now take note that this tRNA over here, the one that's shown over here, is now broken free. It doesn't need to hold on to the methionine anymore because methionine has been bonded through that peptide bond to proline. So now this tRNA, if you watch, it's going to move out and it's going to pick up another methionine. Meanwhile, serine is coming in with its amino acid with its corresponding anticodon. So now we see serine coming in, we see methionine over here joining up with that same tRNA. It's important to note that the tRNA with the code UAC only ever carries methionine. That allows it to be specific so that this code down here that we see, this mRNA code, will always code for the same protein every single time. If these tRNAs just grabbed any amino acid they wanted, there's no way that this code would produce the same protein over and over and over again. So now we're ready to form another peptide bond between proline and serine, and this process is just going to repeat over and over and over again until the entire chain of the mRNA has been turned into a length of amino acids. Now, this is not a completed protein. What we have now is we have a length of amino acids that are linked together through peptide bonds. They need to physically fold up to take a 3-dimensional shape so that they can become a protein. Once they have that shape, then they can go off and do whatever their job is. So again, in recap, what we're seeing here is the 2-step process of taking DNA and turning it into proteins. The first step happens inside the nucleus, and it's called transcription. DNA is turned into mRNA, or messenger RNA. That messenger RNA leaves the nucleus, heads out into the cytoplasm. Then with help from a ribosome and some tRNAs, amino acids are linked together, peptide bond by peptide bond, to form a protein, which then folds up and does its job. And that's our look at protein synthesis.